Protozoology is the branch of biology which deals with the study of eukaryotic organisms belong to the kingdom Protista. Veterinary protozoology is the branch of parasitology which deals with protozoan causing diseases in animal. Anton Leeuwenhoek is the father of biology. Protozoa. Protozoa is the subkingdom of the kingdom Protista, and the term protozoa is coined by Goldfuss. And they are unicellular eukaryotic organisms, and they are microscopic in nature. First protozoa is discovered by A.V. Leeuwenhoek. There is difference between protozoa and rickettsial organisms. Protozoa are eukaryotic, while rickettsia are prokaryotic. They possess cell wall and organelles for performing different metabolic activity, but rickettsia, they are more like a bacteria and divide up cell wall. There is clear-cut difference between the protozoa and protophyta. The type of cell, the protozoa is eukaryotic, but the protophyta is prokaryotic. And the both cells are unicellular animal, that is protozoa, and protophyta is unicellular plant cell. Cell boundary. Protozoa has no definite cell wall, and in case of protophyta, it has most rigid cell wall. And cell wall is made up of cellulose. And the size, individuals of the same species may differ in shape and size, and individuals of same species have uniform shape and size in case of protophyta. Nucleus is well defined in the protozoa because it is eukaryotic, and in case of protophyta, nuclear material is often dispersed in the cytoplasm because this is prokaryotic cell. Cell division is the binary fission, usually longitudinal, and in some case transverse binary fission can be seen, and in case of protophyta there is always transverse binary fission is seen. And nutrition is the most important. In case of protozoa, nutrition is holozoic. In case of protophyta, nutrition is holophytic. Holophytic is because of the presence of chlorophyll, and in case of protozoa there is no utilization of chlorophyll except in case of euglena. Now let us come to the structure of protozoa or an eukaryotic cell. The structure of the protozoa is referred as organelles and not organs, because organs are composed of cells and organelles are differentiated portion of a cell. Basically, protozoa is composed of the mass of protoplasm consisting of one or more nuclei and cytoplasm. Now, first let us discuss about nucleus. It is tree nucleus enclosed in the membrane as seen in eukaryotics. But in case of prokaryotics, there is the primitive nucleus and which is not enclosed in the nuclear membrane. Normally, protozoa contain only one nucleus, that is uninucleates, sometimes binucleate or more multinucleates. Nucleus has nuclear membrane, bounds the nucleoplasm, chromatid and intranuclear body. Now let us come to the types of nucleus. There are mainly two types of nucleus. The first one is vesicular and the second one is compact. In case of vesicular nucleus, this is observed in all the protozoans except ciliates. Vesicular nucleus has nucleoplasm and nuclear membrane. Nuclear membrane binds to nucleoplasm chromatin. Nucleoplasm chromatin is pulgin, that is has DNA, intranuclear body or endosome, which is more or less central in the nucleoplasm, which means in case of nuclear classification there is two nucleus, one is vesicular, other is compact. In case of vesicular nucleus, there is the pulgin positive nucleus. This is because it has DNA in the intranuclear body endosome. Inside the endosome there is the DNA in case of vesicular nucleus, and there is less amount of chromatin. Now let us come to the compact nucleus. Compact nucleus contain large amount of chromatin and small amount of nucleoplasm, and this is observed in case of ciliates. Again, ciliates are classified as two nuclei. One is micronucleus, other one is macronucleus. Micro means which is small and diploid and which divide mitotically. And macronucleus is relatively large and which is polyploid and which divide mitotically. Now let us come to the types of vesicular nucleus. There are again two types of vesicular nuclei. First one is intranuclear body is endosome, which is also called as karyosome, which is divide of DNA, hence it is called as Fulgen negative, in which chromatin which form the chromosome lies between the nuclear membrane and endosome. In case of other type of the vesicular nucleus, the intranuclear body is nucleolus, which is the Fulgen positive which contain DNA and which has one or more nucleoli are present, and the chromatin is present throughout the nucleus. But in case of the first one, that is in which intranuclear body is endosome, where chromatin is lies between the nuclear membrane and the endosome. But in other type of vesicular nucleus, that is the intranuclear body is nucleolus, in which chromatin is present throughout the nucleus. The examples for this type of vesicular nucleus in which intranuclear body is nucleolus is apicomplexa, and in case of intranuclear body is endosome, and the example for such type of vesicular nucleus is trypanosomes and parasitic amoeba. Now let us come to the cytoplasm. This is an extranuclear part of the cell which consists of outer dense hyaline homogeneous layer called ectoplasm and the inner more voluminous fluids or granular endoplasm. The outermost part of the cytoplasm is called as ectoplasm and the innermost is endoplasm. And they have carbohydrates, proteins, lipids, salt, metals, and non-metals. The cytoplasm secretes digestive enzymes, pigments, lysines, and toxins. Peripherally, they may present locomotor organelles and associated structures such as cilia, flagella, and pseudopodia. Now let us come to the ectoplasm part of the cytoplasm. This gives the shape, and which is outermost thick layer is the ectoplast, that is plasma membrane or limiting membrane. And if it is the membrane, it is known as pellicle. If it is layer, then it is called as periplast. If the ectoplasm is in the membrane, then it is called as pellicell. If it is the layer, then it is called as periplast. And the functions of the ectoplasm such as protection, locomotion, excretion, respiration, and adhesions are the some of the functions of the ectoplasm. And now come to the innermost part of the cytoplasm, that is the endoplasm, which is granular because of the inclusion bodies, and it contains nuclei, glycogen vacuoles, food vacuoles, contractile vacuoles, and chromatid bodies, glycogen, fat globules, endoplasm reticulum, ribosomes, mitochondria, Golgi complex, lysosomes, and phenosomes. And the functions of the endosome is help in the nutrition and reproduction. Now let us come to the locomotion in the protozoa. It is done by the majorly 3 to 4 structures such as flagella, cilia, pseudopodia, and other mechanism is gliding mechanism. Now first let us discuss about flagella. Flagella is the whiff-like structure which is composed of central axoneme and outer sheath. This is an outer sheath and central axoneme which arise from the kinetosome of the cytoplasm. This point we have to remember, flagella is arise from the kinetosome of the cytoplasm, and innermost central part of the flagella is axoneme and which is covered by outer sheath. Flagellum may be directed anteriorly or posteriorly and may be attached to the body along the side. And the electron microscopic view of the flagella, which shows axoneme is composed of central 2 filaments which is surrounded by 9 peripheral filaments, that is 9 plus 2 structure. Like inside the axoneme there are 2 microtubules and which is surrounded by 9 structures. Now let us come to the cilia. Cilia is eyelash-like organism. It is an organelle which is fine short than the flagellum and which is also arise from the kinetosome and which is embedded in the ectoplasm. and it helps in the function such as ingestion of food and tactiles. Cilia are found in the Ciliophora members. And now let us come to the pseudopodia. It is a temporary locomotor organelle that can be formed or retracted as needed. This is present in case of amoeba. And there are four types of pseudopods. The first one is lobopod, which is relatively broad pseudopod with dense outer layer and more fluid in the inner zone. This is seen in the amoeba. And now let us come to the phylopod, which is slender hyaline pseudopod that tapers from its base to its pointed end, which tend to anastomose and may fuse locally to produce thin cytoplasm. Now let us come to the mixopod, and which helps in the trapping of food and locomotion, and axopod, which is the projection from the body which has no branchings, that is anastomosine. And the last one is gliding locomotion, in which body glides smoothly along the changes in shape and size, which is appreciable under the light microscope, which is seen in Toxoplasma and coccidian protozoans. And in next video, we will discuss about nutrition in protozoans. Thank you.